Invisible allies have played a decisive role in generating the rich environment that supports human life on Earth. All visible life forms evolved in intimate proximity with the world of microbes and benefit from engaging in multiple symbiotic relationships with them. The European research project SynBio4Flav explores the potential of microbial communities for alternative means of production in response to the massive exploitation of natural resources by conventional production practices. The project focuses on the biosynthesis of complex chemicals, in particular flavonoids, valuable substances that naturally occur in plants. SynBio4Flav mimics the natural process of flavonoid synthesis within plants, enabling Synthetic Microbial Consortia to produce flavonoids that are chemically identical to those of plants.
The revolutionary in this project is that we are trying to change completely the paradigm in the synthesis of complex chemicals.
The difference is that we work at the nanoscale, and our machines are biological elements, enzymes and microorganisms.
I think this project is like a small step towards a more circular and more sustainable economy and way of producing compounds. It's not only true for flavonoids or related structures, but in theory this is applicable to all kinds of both bulk and fine chemicals and molecules.
SynBio4Flav envisions a shift from plant-based sources towards microbial production for a wide range of applications, with a special focus on the health and food sector.
Flavonoids have various health-promoting but also taste-balancing effects, like naringenin is used for bitter masking properties.
It has been shown that these compounds are implicated in prevention of several chronic diseases such as cardiovascular disease, obesity, type 2 diabetes.
When flavonoids are extracted from plants, the yielded quantities are very small. The potentially rising global demand for flavonoids may not be met by sustainable forms of cultivation. Large-scale agricultural production involves extended, water-intensive growth areas, resulting in considerable transport and waste volume, with related environmental challenges and costs to be taken into account.
Our main interest is to get an enhanced production levels of the final flavonoids using this biotechnological approach, this novel biotechnological approach.
Microbial production processes are well-established technologies that have been applied for thousands of years to make cheese, bread, beer and wine, and many other culinary specialties that are part of the local tradition of all cultures worldwide. But microbial production holds a potential beyond the transformation of plants and animals into food.
It's fascinating how microorganisms can turn any kind of feedstock into something valuable. And this is coupled to the ability we have developed in the last 70 years to really engineer those systems.
Synthetic biology is a rapidly advancing discipline that opens up a huge space as an alternative production system for traditional as well as novel applications. It's fueled by groundbreaking developments such as CRISPR-Cas9, a Nobel Prize-winning genome editing tool derived from a natural bacterial defense system against viral attacks.
It is important that people get informed on new developments in biotechnology, in synthetic biology, because it may revolutionize our future life. We encourage dialogue on the rapidly advancing new fields, such as synthetic biology, through our podcast mini-series Made by Microbes, and through our conversations on metabolic engineering with different experts from different fields.
To achieve its ambitious goal, the project builds on the expertise of 11 European project partners involving synthetic biology, systems biology, biochemistry, molecular biology, bioinformatics, and chemical engineering, developing two main concepts.
The first is the distributed catalysis, so the concept of split a complex biotechnology pathway in the minimal functional module that we can optimize separately and then put together in order to resemble the original pathway in the context of a synthetic microbial consortia. This is a very important concept.
And it's the consortium idea, because traditionally, if you think about producing chemicals, products with the help of microorganisms, you have a single species, a single microorganism that does the entire job. So what we have here is this microbial community, the consortium, that has divided its tasks. So every part of this consortium has its special role, and together they act then in the benefit of producing chemicals.
In our approach, each one of them enzymatic steps for getting the final compound, each one of the reactions is separate in different species, in different cells. E. coli, Pseudomonas putida, Streptomyces albus, Saccharomyces cerevisiae.
The microbial production of flavonoids in consortia, and in fact its modularity, allows us to precisely design the process to obtain a specific product.
The other concept that we are developing is the concept of standardization in biochemical production. This means that we can combine modules and reuse parts, saving time, standardizing the synthesis of complex chemicals.
If you have a more modular system, you can use it in a plug-and-play manner, which means you can also adapt faster to changing requirements.
The project contributes to standardization on many levels through the continuous improvement of libraries and tools such as the bioengineering platform Dulix.
One of the major problems we have in turning ideas into products is that we are really missing the data required to shortcut product development. Dulix specifically addresses this issue by building a fair, compliant database of sequences and related data.
Experimental work in labs provides the reference data that serves as the standard for quality assurance.
The company has a long history and expertise in extraction, synthesis, biosynthesis, analysis of natural products, and we provide what is called analytical standards of the substance of interest in the research conducted by the consortium. So we have offered a collection of about 80 different molecules to support the research of the other partners.
Lab automation and computer models were applied in iterations to select the most promising strains and strategies for further optimization. Lab-validated data was fed into computer models to tackle the complexity of metabolic networks, not only simulating the activity of a single enzyme, but also the behavior of an organism and the Synthetic Microbial Consortium as a whole.
We have developed a machine learning algorithm that can tell us how efficiently and how fast an enzyme works. So traditionally, you would have to test this in the lab, which can take days or even weeks. With help of this machine learning algorithm, you can do that in fractions of a second. So it is a really useful tool to simulate enzyme activities and help designing engineering strategies for our microorganisms. What we've also developed is yeast strains that tell us how much they produce with the help of so-called biosensors. They work with the output of fluorescence. The higher the fluorescence, the more the yeast cell produces of our product of interest. So that makes it very easy for us to pick those cells that are the most efficient producers if we want to select those.
Integrated robotic systems have been used for screening samples at high production rates and accuracy, replacing repetitive manual tasks in labs, supporting data collection and documentation. Reducing the workload of the scientists, it allows them to concentrate on their core competence of interpreting results and designing new experiments.
We developed a very efficient high-throughput screen method combined with a robot system and microfluidics.
Research results have been verified by in vivo experiments with animal models and optimized within bioreactors of increasing scale to approach industrial upstream as well as downstream processes.
We optimized the fermentation process of the strain E. coli, Putida, Streptomyces albus, Saccharomyces cerevisiae at laboratory scale. Then we continue with the scale at pre-industrial scale in bioreactors of 1.5 liter, 5 liter.
The European Union is putting a lot of effort, also economic, in a way that different European companies, research organizations and universities are able to provide to the society flavonoids in large quantities, which are important pharmaceuticals and nutraceutical compounds for the society.
By offering this sample of new molecules or rare molecules to the market, gives the opportunity of academic research or industry to make activity studies and to work on the utilization of these molecules for different applications. In my opinion, it has a great potential in terms for the industry.
By the end of the project, in August 2023, SynBio4Flav successfully provided the proof of concept of positive interactions between different microbial species, driving efficient bioproduction within Synthetic Microbial Consortia. In addition, the project has played a significant role in standardization within synthetic biology and the adoption of computer-assisted design methods for complex chemical processes. The ambition of the 11 partner organizations behind SynBio4Flav extends beyond the production of flavonoids. Through variations in the composition of synthetic microbial consortia, microbial cell factories have the potential to produce virtually any complex organic substance, thus delivering a sustainable, cost-efficient alternative to traditional extraction from crops. Indeed, microbial biomanufacturing promises to become a key player in meeting the needs of a growing world population and at the same time enabling the preservation of precious natural resources. This project has received funding from the European Union's Horizon 2020 Research and Innovation Programme under Grant Agreement number 814650.